All right, everyone, let's do a little example of transcription and translation by playing around with a provided DNA sequence and figuring out what the protein is that it's trying to encode within its genomic region. So a few disclaimers. We are going to assume that the DNA sequence that we're looking at is going to be an entire gene, and we're also going to assume that this is going to be the template strand that is going to allow, remember, our RNA polymerase to move in the fact that it will be adding onto the 3' end of our newly synthesized RNA strand. So the first thing we're going to do is we're going to go ahead and take our DNA and we're going to complete transcription. And that means that we're going to have to take a look at our individual provided nucleotides and match them with their complementary RNA partners. Remember, G gets matched with C, A will get matched with U, and if you come across a T in DNA, you will match that with an A in RNA. Now you also have access to this PowerPoint in your D2L file, so please make sure that you can put it either in slideshow mode, so as you click through it, you'll be able to see the same exact steps that you're seeing on the video, and you can obviously also work out the problem as you follow along to the video. Okay, so let's go ahead and let's complete our first step. All right, so we need to determine what our complementary RNA strand is. And this would be a good time to kind of pause the video. And on your end, you're basically going to match all the different letters to their complementary pair. So G gets matched with a C. So imagine that the RNA polymerase is reading these nucleotides on the template DNA, and it's matching it to a complementary RNA. So G gets matched with C, A gets matched with U, T gets matched with A, C with G, and just do the entire strand. So pause the video right here, and then whenever you have your whole strand matched up, go ahead and hit play again so we can compare answers. All right, so this was my RNA strand. This is my little RNA that I produced. So notice how there's no T's, right? Because RNA doesn't have T, it only has U's. So this is my RNA strand, and we're going to make another assumption. We're going to assume that our RNA is going to be fully stabilized, so it has a cap on the 5' end, and it has a poly A tail on the 3' end. We're going to assume that the entire strand is an exon, meaning we're not getting rid of any pieces, which means that we're going to go ahead and virtually export this RNA molecule from the nucleus into the cytoplasm, and we can go ahead and complete our second step, which is to take our strand of RNA and go ahead and break it down into sections of 3 that will represent our codons. So let's go ahead and let's divide up our RNA into codons. Here you go. So all I did is I took my same strand and I just made a little mark to separate the 3 individual letters. And we're going to assume that the reading frame will start with the first letter, and then we just count down, and after every 3rd letter we break it down and we start a new codon. Now once you have your codons established, this is then where your codon table will come in handy, because remember, we're now looking at mRNA. So we are going to play the role of our rRNA and read the codons and figure out which amino acids we need to bring over. Now, an important thing to remember is that we have a start codon and we have a stop codon. So let's go ahead and let's locate our start codon. Hopefully we're all thinking of the month of August. And I'm hoping that right now everybody's saying it's right over here. You got it. Now it's important to recognize the start codon because we don't care about anything that happens before the start codon. Okay? So here you go. So we located our start codon. And then now that we have our start codon, we can go ahead and start continuing on reading the rest of the codons. All right, so use your codon table, which is provided on the next slide on your end. And what we're going to do is, since we've already identified the start codon, we're going to go ahead and we're going to keep reading our codons until we reach a stop codon. Okay, so go ahead and reference the codon table. I'm going to pull it up on my end. Here's my little codon table. It's a little bit small. You have a larger one in your file. And like I said to you before, start codon is AUG. We do not have to write down the amino acids, the methionine, because oftentimes it will be cleaved away. So I just kind of like to circle my AUG to indicate that this is my start. If you want to write down methionine, you can. If you want to abbreviate it to MET, you can as well. But either way, remember, we don't care about anything before the start codon. So just ignore that particular section. Let's go ahead and take a look at the first codon we encounter after the start, which is going to be GGC. Remember, we're going to read the table starting on the left and going across. So locate your G. There you go. Go across. Find your second G, which is all the way in this box right over here. And we're going to do GGC. And that's going to give us glycine. So I'm going to go ahead and abbreviate the glycine GLY underneath it so I can keep track. And I'm going to move on to my next codon, CGU. Find your first letter, C, go across, G is in my last box right here. And I'm looking for CGU, and that's going to give me arginine. So ARG. Alrighty. Now, if I'm going too fast for you, feel free to stop the video so you don't feel like you have to keep up with my speed. So you can stop the video and then go ahead and look for the next codon. And then whenever you're ready, come back and then we'll compare if we found the same one, okay? Next one is ACC, proline, PRO. All right, and then the next one is U. Here's my U, going across to A, UAA. Look at that. That's my stop codon. Remember, the stop codon does not get an amino acid. That means that we have reached the end of our translation, which means we don't care about anything that happens afterwards. So I scratch it out, but you can just ignore it. You don't have to worry about it. So thus far in our provided protein sequence, we were able to go ahead and match it to the complementary RNA. That was the transcription step. Then we divided everything into codons. We found our start codon, AUG, and then we started reading our codons using our table, and then we stopped when we reached one of the stop codons. Okay, so now let's go ahead and let's just clean this up a little bit, because I'm going to go ahead, and this is what you're seeing on your end. So if you can't read my little chicken scratch, that was the amino acids that we found. And then what I would do as a final step is I would clearly indicate what my peptide strand is. And we do that by writing down the amino acids, whether you write down their whole name or their approved abbreviations based on the codon table. And you want to only write down the amino acids between the start and the stop. If you want to write start and then write the amino acids and write stop, you can. By all means, that's okay. If you just want to do what I did, which is I wrote the amino acids that are in between the start and the stop, that's fine as well. But what you should absolutely not do is tell me what these amino acids are and what these are. You should not be writing down anything that happens before the start and anything that happens after the stop, okay? You only care about what's in between, because that is going to be your translatable area. That's the area that you are actively collecting your amino acids, and when it says stop, we stop. So at the end of the day, you now have this beautiful sequence of amino acids. Obviously, in our body, it would be a much longer amino acid sequence, and it would then go into its modification step, which will involve the use of things like our chaperone proteins to reach their final shape. For your exam, I will be giving you a new DNA sequence, and I will be asking you what protein it is be able to produce. So I will provide you with the codon table. In fact, let's click over to the next slide. This is the exact codon table that you're going to get, with the exception that, as I mentioned in the lecture, I'm going to erase the start. So yours will not say start. It will say methionine, and remember, it will be up to you to memorize the fact that your start codon is AUG. So once you have your DNA, make your RNA, divide it into 3, find your AUG, and then keep reading until you reach your stop. And then your final polypeptide, or your protein strand, will be the amino acids that you've collected between the starting point and the stop codon. All right, so go ahead and play around with this. If you have any questions, please do let me know. And I hope you enjoyed this little example.